All right, everyone, we are live. A very good afternoon and warm welcome to Sri Chaitanya channel. Everybody, I welcome all of you, all the NEET 2024 aspirants. Basically, these classes are organized specifically for you people, wherein we are going to pick 25 most important topics per subject, and daily you will be having classes. And the series is NEET 10, which is the game changer series. Why NEET 10? Reason being, this is for the last 10 days. Why game changer? Supposingly, you are at 500 marks right now. So just by solving these questions in all the sessions, botany, zoology, physics, chemistry, your marks for sure will be 50 marks up from wherever position you are right now. All right. So without wasting further time, let's get started. Let's start our session, and before starting, everybody do hit the like button and share it with all your friends. Don't forget to subscribe our channel also, because you will be notified about all the lectures that we are going to conduct. Good afternoon, Dr. Queen. Good afternoon, Rahul. Hello, Sana, Angel. Good afternoon, Maneem. Good afternoon. So all right, everybody, let's get started. This is a game changer series to score 650 plus marks in your NEET examination. Your NEET exam is just after, I would say, 7 days only. Reason being, from 1st of May onwards, you will be organizing a lot many things. So we are left with these 7 days only. We have to double the practice so that we can ace our examination, so that we can ensure that we are going to get a government medical seat. All right. Ravi Yadav, you're getting 550, okay. If you're getting 550, then of course your marks will be up, but, there is a big but, which is you have to analyze all the tests that we are going to conduct. See, supposingly we are going 25 questions in each and every session, then do finalize like which questions are correct, which ones are wrong, which question you haven't attempted. Okay, so keep a note of all those questions that you are not able to attempt, or the questions which you gave the wrong answer. Now the actual picture begins just after the class, wherein you are going to reread, you are going to revisit all the concepts from which the questions were wrong. Okay, by analyzing all those concepts, by reading all those concepts, going to the next class just after doing this exercise only, then only you'll be able to increase your marks for sure. All right, Ravi. So I hope from the previous class, that was yesterday, everybody has revisited all the concepts that of the yesterday class. Everybody? Total how many chapters will cover, Chandana? We are going to cover all the chapters, be it 11th, be it 12th. You're going to get questions from the entire syllabus, because this is the time wherein we should practice from all the chapters, be it 11th class, be it 12th class. So you're going to get questions, rapid fire questions, rapid fire 25 questions will be there from 25 different topics. One thing I can ensure that these 25 different topics that I have picked up are thoroughly researched. These are the topics, if you will solve the PYQs, you're going to see that these concepts are there in all the PYQs of last 10 years. So I have deliberately picked up all those concepts that are repeatedly coming into your examination, and from those concepts only I have framed the question, and we are going to practice such questions on daily basis on this very beautiful channel. So on that mark, do hit the like button and subscribe our beautiful channel. Yes, all right. So let's get started, everybody. If I give you the classes timing, your timing starts from 12 up to the 5:30. This is Veena Yadav, your botany mentor. So from 3 to 4, our time has been booked. So from 3 to 4, mark your calendars, and you have to come on daily basis from 4:30 to 5:30. Pawan Kasu sir is going to conduct your class for chemistry. All right. On that note, let's get started with our very first question, kids. This is the first question for today's session. This question is from the very first chapter of 11th class, which is Living World. From Living World, 2-3 topics have been removed in the latest syllabus, which are your taxonomical aids. All right, but this particular topic is still there in the Living World. All right, you have to answer in the chat box and be super quick, everybody. Bhavani Prasad has answered. Renuka has answered, Dr. Queen has answered, Kiran Era has answered, Love Ramana has answered. On the Goshala channel, I can see Ravi Yadav. Yes, please answer. Charan has answered, Anjana Reddy has answered. Perfect. The question is, which one among the following is a defining feature of all the living beings? If I speak about living beings, we can have number one defining feature, number two we can have characteristic features. What is the difference between these two? So if I want to keep a specific property into defining feature, it should fulfill two criteria. Number one, it should be present in all the living organisms without any exception. There should not be even a single exception at all. And number two is, it should not be present in any of non-living entity. Yes, you heard me right. So if any criteria is basically fulfilling these two conditions, then it will be defining feature, else it is characteristic feature. So if, let's say, some properties is in the most of the organisms, not all the organisms, there are certain exceptions also, then we will keep it into characteristic feature. If, let's say, any property is found in both living as well as non-living things, then we will keep it into characteristic feature again, okay? So, in this question, we are asked about the defining feature. So, growth, first of all. Growth is also found in the case of non-living entities. Yes, everybody? In the non-living entities also, it cannot be defining feature since we can see growth in non-living. However, the growth in case of non-living is extrinsic. It is extrinsic growth, right? But there is growth, which means it cannot be defining feature. Number two is reproduction. So there are certain living organisms which are not able to reproduce. Examples, mule. Examples, worker bees. Example, there are certain sterile couples also which are not fertile, which cannot produce babies, okay? So we do have exception in the case of living organism. Okay, exceptions in the case of living organisms, which means this cannot be the defining property, correct? Number C is metabolism. So metabolism is basically sum of all the reactions which are happening inside our body, be it anabolic reactions, be it catabolic reaction. So sum total of all the reactions which are happening would be metabolism, okay? And it is found in all the living organisms without any exception. Secondly, it cannot be found in any non-living entity also, which means it is able to fulfill both the criteria of defining feature, which means we can say it is a defining feature. Option number D is a self-consciousness. Students, consciousness is found in all the living organisms, but we are the privileged ones. Human beings do have self-consciousness also. Consciousness simply means awareness about your surrounding, but self-consciousness means not only you are aware about the surroundings, but you can analyze all the things. You are self-aware about yourself. Supposingly, people are studying right now, you are having this thing in your mind that we are going to become doctor very soon. This is what? This is self-awareness. If we will study, we will become doctor. Our life will be set. Okay? So this is called as self-awareness or self-consciousness. So human beings has got self-consciousness. Rest all the living organism has got consciousness. So self-consciousness cannot be a defining property. Out of these four options, your metabolism is the only one which is defining property. But reactions occurs out of the living body in the test tube. Yes, of course, reaction do occur outside the body in the test tube. Those are your live reactions. Okay, your metabolism needs a cell. Within a cell, if the reactions are happening, then that is under criteria of metabolism, okay? Outside the cell, in the cell-free system, in the laboratories, all the chemical reactions which are happening, we cannot consider them, okay? Rasagna? All right. So if your answer is option number C, everybody shout out, give me shout out with +4, +4 kids, +4 kids. I can see lot many answers which are correct. Charan, Anjana, Ramana, Naju, Vikram, Pillai, +4, Moonshine, +4, +4. All right, moving on to the next question then. Very interesting question. And it was an ambiguous question of PYQ only. Renuka, perfect. Next question. Next one. Which one of the following is not, please pay attention, this is a keyword, not a main criteria for classification used by Whittaker? If we speak about R.H. Whittaker, sir, who gave us 5 kingdom classification system, according to which we have 5 different kingdoms: Monera, Protista, Plantae, Fungi and Animalia. All the living organisms were put into these 5 different kingdoms. Yes, I'm getting the answers perfect. Varshini, Kiran, very good. Anjana, very good. Moonshine, very good. Uttej vlogs, very good. Dr. Queen, Karuna, Neer, good afternoon. Yes, yes. Perfect, everybody. So, maximum of you have done it correctly. Cell membrane is basically not the main criteria. Don't get confused. See, in NCERT, we have this one table in the NCERT wherein your Monera, Protista, Plantae, Animalia and Fungi has been written, and there are certain differentiating features which are given. Okay, so don't get confused. Those are basically the differentiating features among all your kingdoms. Those are not the main criterias. Main criterias are separately written in NCERT text itself. Okay, so don't get confused between the table and the text. The table is giving us the differentiating features of all those kingdoms which are Monera, Protista, Plantae, Animalia and Fungi. But in the text, in the NCERT text, it is clearly mentioned that main criteria are cell structure, mode of nutrition, reproduction, phylogenetic analysis and body organization. So cell membrane is not the criteria. That is why it will be the correct answer. Alright everybody, option D is +4. Option D, students with option D +4, can I see +4? Everybody, Ramaya, Karuna, Charan, Rupa, Renuka, Neer, Naju, Uttej +4, Y +5, +4, yes, perfect. Rajya Lakshmi +4, Rasagna +4, Rakshana +4, perfect. Moving on to the next question then. Dr. Papu is dal, I guess, yes? Next question, please. We have 2 lists, list 1 and list 2. In list 1, we have archaebacteria, chemosynthetic bacteria, cyanobacteria and heterotrophic bacteria, right? Then list 2 has some properties related to all these bacteria which are written over there, right? So in the list 2, we have most abundant bacteria, number 1. Heterocytous bacteria, which means they do have presence of heterocyst. Number 3 is oxidation of various inorganic substances. And number 4 is bacteria that live in harsh conditions. Come on everybody. I can see a lot many correct answers. Uttej, Varshini, Moonshine, Ramaya, Neer, Kiran, Naju. Perfect. Anjana, Rupa, perfect. So the correct answer is option number B, as most of you have mentioned. See, if I speak about this question, archaebacteria number 1. So in bacteria, there is segregation of eubacteria and the archaebacteria. Eubacteria basically is your true bacteria, okay? And archaebacteria is the one which thrives in extreme conditions. What do I mean by extreme conditions? Extreme simply means very high temperature, acidic conditions. They are methane gas producing. So these are the extreme conditions. So my archaebacteria thrives and survives into the extremities. But my eubacteria is the true bacteria surviving in normal conditions, all right? So, archaebacteria first of all, okay, answer is option number 2. Archaebacteria, bacteria that lives in the harsh conditions, all right? Then chemosynthetic bacteria. So, we can have photosynthetic bacteria and chemosynthetic bacteria. Basically, both these bacteria are able to synthesize their own food. But the difference is, for the formation of the food, we do need ATP. If the source of ATP is sunlight, then we call it photoautotropic. If the source of ATP is from any chemical reactions, then we call it chemosynthetic bacteria. Okay, so based upon the source of ATPs, we have segregated, we have divided this into two different types, correct? Now, chemosynthetic, which means oxidation of inorganic substrate, all right? Then we have cyanobacteria, number 3. Now, cyanobacteria, they do have two properties. Number 1, of course, they can photosynthesize. Number 2, these are the ones which are able to fix nitrogen. Now, in order to fix nitrogen, we do need nitrogenase enzyme. And the catch is, nitrogenase enzyme is able to function in a proper way when there is anaerobic condition. But in case of photosynthesis, there will be aerobic condition. Now, there is the need of the hour that we need to make a separate compartment for photosynthesis, separate for nitrogen fixation. In order to achieve this, we have normal vegetative cells. Excuse me. Sorry. Yes, in order to achieve that, we have normal vegetative cells which are undergoing for photosynthesis, and we have heterocysts. They are basically responsible for nitrogen fixation, all right? So in heterocyst, my nitrogenase enzyme is present. And how come my nitrogenase is able to function in an efficient way? Because in heterocyst, I have maintained anaerobic conditions. Anaerobic, which means in heterocyst, photosynthesis is not happening. All right, so cyanobacteria is heterocyst bacteria. Then heterotrophic bacteria, although bacteria are nutritionally very diverse, but majority of the bacteria are heterotrophic in nature. So option D, let's match it with option number 1. So A is 4, B is 3, C is 2, and D is 1. Answer will be option number 2. Yes? +4 everybody? +4, can I see +4? +4 everybody? Yes. All right, perfect. Moving on to the next question then. +4, Uttej, +4, Karuna, Kiran, Renuka, very good. Okay, in the last video, you people didn't told me about the homework question number 1, and you people didn't told me about your marks also. So in today's video, I really want from you guys, in the comment section, do write your marks of today's session, as well as in the last, I will be giving you a homework question. Please solve that question also. Okay, is that a promise, everybody? Is that a promise? Yes, ma'am, this is a promise to you. Come on, next question then. Next question, kids. Navya? Okay. Next question. Hydrocolloids, algene, carrageen are obtained from? Very famous question. Very famous question of NEET exam. There are certain hydrocolloids like algene and carrageen. Your NCERT clearly mentioned. Kiran, Varshini, Renuka, great. Rest of the kids? Okay, meanwhile, please do hit the like button also and subscribe our beautiful channel so that we can keep giving you such beautiful classes. Navya, great. Naju, great. Rest of the kids, come on. Maneem, Rajya Lakshmi, can I see your answer? Dr. Queen, perfect. So, algene and carrageen, they are obtained from, we know it, algae. But which algae? From brown and the red algae respectively. From red algae, we are able to obtain one hydrocolloid substance called as carrageen. From brown, we can obtain algene. Okay, the correct answer will be option number D, which is brown and red algae respectively. All right, everybody is correct. Please put a +4. +4. Ma'am, can I give NEET in 11th grade? No, I'm afraid you cannot give NEET exam when you are in 11th grade. Just after your 12th only you'll be able to appear in the exam. But yes, there is no upper age limit. After appearing for your 12th class, you can give NEET multiple times. Okay, there is no upper age limit attached to it. Okay, +4. Very nice. Moving on to the next question then. Next question, kids, on your screen. This question is from morphology. How many of the given main primary functions of the roots? So basically, these 5 functions, we have to identify which ones are the function of the main root and which ones are not. Hello, King Koshal. Please attempt the question. Come on, everybody. If I speak about roots, how many are the primary functions of the root? Number 1 is absorption of water and minerals. Number 2 is nitrogen fixation. Okay. Number 3 is proper encouragement to the plant. Okay. Adherence. They are providing adherence. Number 4 is storing reserve food material. Okay. Synthesis of growth regulators, which are your plant growth regulators, be it your auxin, be it your cytokinins. Great answers, Dr. Queen. Uttej, Navya, Moonshine, Rakshan, Rupa, Karuna, Manim, Rajalakshmi, Renuka, Neer, perfect everybody. Honey Reddy, very good. Very good, very good. All right, so we have these 5 functions which are written over here. If I speak about the first one, absorption of water and mineral, of course, it is a function of root only. And if I may ask you the second question, specifically, which part of the root is responsible for absorption of water and mineral? Can you guys tell me? Specifically, which part of the root is responsible for absorption of water and mineral? Can you tell me, guys? Which part specifically? Come on, everybody. Specifically, which part is responsible? Yes, it is root hairs, because root hairs are present on the area of maturation, and the function of root hairs is basically to increase the surface area so that we can absorb more amount of water. All right. So first of all, yes, this option is perfectly correct option. Then nitrogen fixation. Every root is not able to fix nitrogen. If the bacteria is attached to the root, then of course they can go for nitrogen fixation, but it is not the function of the root. If this would have been a function of root, every plant naturally would have been able to fix nitrogen, but it is not like that. So this is your incorrect option. Then proper encourage, which means attachment to the plant. All right, my plant is having soil, right? Roots are below the soil generally, so roots are providing encouragement or the attachment point for the plant, right? They are basically holding the plant upright in the position. So this is also correct option. Then storing reserve food material, of course, correct one. And they are also able to synthesize growth regulators, which are your phytohormones. So 1, 2, 3, 4. 4 statements are correct with respect to roots. 1 is incorrect. That is why answer will be option number C, which is 4. All right, everybody, plus 4. Can I see plus 4? Plus 4, everybody. Plus 4. Yes, Rahim, correct answer. Pal Sahib, correct answer. Honey, correct answer. Risagna, correct answer. Rajalakshmi, perfect. Correct. Yes, perfect. Moving on to the next one. Come on, next question, everybody on your screen. Okay, meanwhile, if you are a student of NEET 2025, so Vedya batch is already live on all our platform. Vedya batch is basically for all the dropper students who are going to appear in NEET 2025. Okay, Vedya batch has got these proven strategies. See, if I speak about Sri Chaitanya, we have 29 years of legacy. Like every year, toppers, rankers are coming from our institute only. So basically, the kind of study material we do have, the kind of educators we do have is very, very different, I would say. Okay, so try it out. There is this Google form in description box. Please fill that Google form so that we can tell you more about this amazing batch, which is Vedya batch, and the seats are limited. All right. Yes. Naju, perfect. Nazanin, perfect. Uttej, perfect. Moonshine, Varshini, great. Anjana, very good. Which of the following is an endospermic seed? Which is endospermic seed? It is your castor. Everybody is correct. Castor. Yes, it is for droppers, Dr. Queen, the students who want to appear in NEET examination in 2025. It is for those students. Okay. Yes. Option number D. Yes. Rasagna, very good. Rajalakshmi, perfect. Plus 4, everybody. Plus 4 in this question. And moving on to the next question then. This question is from anatomy. Next question, everybody, from anatomy. Which of the following is the primary meristem? Which is primary meristem? Okay, primary meristem is basically the meristematic tissue which is present from the beginning itself. Okay, secondary meristem basically appears in the plant life in later life, but primary meristem is from the beginning itself, right? So from the beginning, do we have apical meristem? Do we have intercalary meristem? Do we have cork cambium? Do we have interfascicular cambium? Please pay attention. It is interfascicular, not intrafascicular. Please pay attention. Very good, Moonshine, very good, Rupa, very good, Uttej, very good, Naju, Vinama, Dr. Queen, perfect, perfect. Pearl Sahib, Rasagna, very good. Rajya Lakshmi, very good. It is option number D. See, core cambium is basically your secondary meristem. Why so? Reason being, we have cortical cells. When my plant is going for secondary growth, in that case, the cortical cells will modify themselves and they will make core cambium. Okay, so initially it was cortical cells, which is a permanent cell. Then these cells have differentiated and formed core cambium. So it is my secondary meristem, right? Then interfascicular cambium. This cambium is found in between the bundles of xylem and phloem. Okay, now the thing is, initially there were medullary rays which have modified themselves into interfascicular cambium. Again, a typical case of secondary meristem. So this is secondary meristem, this is secondary meristem, but apical meristem found in the plant at tip of shoot as well as root. So we have root apical meristem in the root, we have shoot apical meristem on the shoot. Okay, so apical simply means the tip. The meristem found at the tip is your apical meristem, present from the beginning itself, right? Number one. Number two is intercalary meristem. Now, we have certain meristematic tissue which is intercalating between the two permanent ones. Okay, so we have two permanent tissues. In between that, we have intercalation of intercalary meristem. Now, what is the function of this intercalary meristem? Supposingly, if I speak about grasses, so this is my grasses, and my browsing animal will come and browse the apical part of the grass. Okay, now there is no more apical part. What will happen? How come this plant will be able to grow now? In that case, we have intercalary meristem which will come into the picture. Now, these intercalary meristem will behave as apical meristem and they will start performing the function of apical meristem. Alright. So, apical and intercalary, these two are the primary meristem present from the beginning of the plant's life. Okay. So, answer will be option number D, +4. Angela, -1. Now, I hope the question is clear to you. I'm really happy to see your honesty here. I hope now this concept is clear to you. Yes? The concept is clear now? So in the final exam, you will do the question from this concept, correct or incorrect? I'll do it. I'll do it, pakka. I'll do it, promise. Okay. Okay then. Okay kids, +4, +4, +4. Yes, we'll be able to do it. +4 in this question then, in the final exam, +4. Dr. Queen, see, intrafascicular cambium is present from the beginning itself. If I speak about dicot plant, so intra is there in between xylem and phloem. But inter is not there. Inter will be formed when your plant is going for secondary growth. In that case, my medullary rays will modify and form interfascicular cambium. Is this clear now, Dr. Queen? Yes. All right. Then moving on to the next question then. Which of the following is a non-membrane bound organelle? There is this one organelle which is membraneless, and that is found in both prokaryotes as well as eukaryotes. Such a simple question and randomly asked question. Such a simple and randomly asked question. We need to find out one membraneless organelle found in both the cases, in prokaryotes as well as eukaryotes. Yes, kids? Varshini, great. Rajya Lakshmi, great. Rasagna, great. Honey, please pay attention and then tell me again. Pal Sahib, great. Uttej, very nice. Chinni, very good. Harshmita, very good. Sheikh, Kiran, very good. Very good, very good. This is your ribosomes. So ribosomes are membraneless and they are found in both the cases, prokaryotes as well as eukaryotes. However, the difference is, in the case of prokaryotes, the ribosomes are of 70S type. But in the case of eukaryotes, ribosomes are of ATS type, okay? So my ribosomes in case of prokaryotes, we're gonna see 70S type. In the case of eukaryotes, we are going to see ATS type. That is the major difference, all right? 70 and ATS. Perfect. Answer will be option number C, +4 everybody. +4. Perfect. Perfect. Moving on to the next question then. Next question. With +4, let's move ahead to the next one. Mitochondria and chloroplast are exception. Yes, Ramala Kaveri, in case of mitochondria and chloroplast, see, these two are the double membrane bound organelles. These two are semi-autonomous organelle. These two divide by binary fission. So there is this theory that we believe that once they were prokaryotes, they started living inside the body of the eukaryotes, and you know, there was symbiotic association. So basically they stayed there forever, okay? Something we call as endosymbiont theory. So according to which, in case of mitochondria and chloroplast, they do have 70S type of ribosome. They have their own circular DNA also, okay? They are capable to synthesize their own protein also, but half of the protein only. Okay, for the rest half, they are dependent upon nucleus. I hope it is clear. Yes? Perfect answers, perfect answers, perfect answers. How many ATP and NADPH are required respectively during C3 pathway to reduce one CO2 molecule? So we are speaking about C3 plants here first of all, okay, and per CO2 molecule. So per CO2 molecule, per CO2, everybody please pay attention. Per CO2, I do need 3 ATP and 2 NADPH2. Basically, 2 ATP and 2 NADPH2 in reduction step, wherein I am making glucose molecule, and 1 ATP in regeneration step. Alright, so thereby in total 3 ATP and 2 NADPH2 has been utilized for Calvin cycle. Alright, Pal Sahib, Ramala, Mani, Sabila, Chini, Rupa, Sure, Uttej, Rakshan, Anjana, Naju, Dr. Queen, Neer. Is this clear? Yes, kids? Is this clear? Is this clear to all of you? I hope you are enjoying the session, because I am enjoying the session a lot. If you are also on the same page, please do hit the like button and subscribe our beautiful channel also. Yes? Yes, yes. +4? Alright, let's move to the next question then. Come on, next one. G1 phase. G1 phase corresponds to the interval between G1 phase. Thank you very much. Thank you. Thank you, kids. It means a lot to me. Come on, next question. Next question, kids. G1 phase corresponds to the interval between what? What is G1 phase? G1 is gap 1 phase. Yes? I'm getting the answers. Thanuj Kumar, Renuka, Varshini, Rupa, Navya, Neer, Uttej, Anjana, Reddy, Naresh, Rajya Lakshmi, Maneem. Come on, everybody. Hashmita. Perfect, perfect. So G1 phase, if I speak about the sequence of the events in case of cell division, basically we have number 1 interphase, number 1 is interphase, and number 2 is my M phase. Number 2 is my M phase, alright. Now, in interphase, we have G1S, G2, right? In M phase, we have M phase. If my cell is dividing again, then there will be G1S, G2M. Perfect? Yes, ma'am. Now the question is asking about the G1 phase corresponds to the interval between, this is my G1 phase. Interval between what? Interval between mitosis and S phase. Yes or no, everybody? Mitosis and S phase. Your question might ask you, it is an interval between mitosis and the phase in which there is DNA replication. There is DNA replication. Okay? So your question might be tilted in this way also. Right? So the answer will be again, you need to understand this sequence. These 3 are my interphase. This is my actual division phase, which is M phase, again interval, again M phase. Alright, so G1 is basically in between my M phase and the S phase. Is this clear? Is this clear? The students who got -1, the concept is clear to you guys? Yes or no? See, for me, it is more important to make you understand the concept behind the question. Because, you know, question is right or wrong doesn't really matter. Just after the session, if you have understood all these concepts, that do matter a lot. Because you're going to get question from these concepts in your final exam also for sure. Yes, ma'am? Is this clear? Okay, perfect. +4, +4, +4 everybody. Moving on to the next question then. Next question. Thank you very much, Dr. Queen. Next question, kids. Select the incorrect match. Incorrect. We need to find out the incorrect statement. All right? So we have different hormones from this particular chapter. Mark my words, from this particular chapter, you're going to get a question from functions of phytohormones for sure. 2 questions will be there for sure from the functions of phytohormones, right? Great. Ramala, Naresh, Anjana, Kiran, Prabhavati, come on, great. Varshini, great. Chinni, great. Utej, great. Dr. Queen, Rupa, Navya, please keep on trying, keep on trying. Come on everybody, keep on trying. Moonshine, Renuka, great. See, so we need to find the incorrect match. The first one is about the auxin. Auxin is a phytohormone responsible for root initiation on the stem cutting. Of course, this is the main USP of my auxin hormone. This is a correct statement, right? Then we have gibberellins. They delay senescence of the fruits. They delay aging process of the fruits. Correct, all right? Then cytokinins, they overcome apical dominance. What is apical dominance? My plant is growing in its this direction, okay? If I want a bushy appearance of the plant, in that case, what I need to do, I just need to cut down all the meristematic tissue which is at the tip part. Have you seen all those gardeners having this big scissors and cutting all these bushy plants from the tip part? They are simply making sure that this apical dominance phenomena should be overcome. Because if there will not be any apical meristem, then lateral meristem will grow and my plant will be having bushy appearance, all right? So in order to get that bushy appearance, we need to overcome the apical dominance done by cytokinin in naturally in the case of plants, okay? Then option D, ethylene, promotion of seed and bud dormancy. It will not promote. It will not perform. Yes, of course, Ramala, in the case of tea plants also, Ramala, you can simply see in any garden also, you know, in the entrance of the garden, there are these beautiful bushy, yes, in hedge making also, in tea plantation also. In the entrance of any garden, you might have seen that entrance is basically with these beautiful bushy plants on the corners, all right? So those are made bushy just by this phenomena only. All right. So answer of this question will be option number D. That is the incorrect match. Hence would be correct answer. All right. +4. Yes, in the parks. Perfect. Perfect. +4 everybody. +4. Okay. Moving on to the next one. What is the function of tassels in the corn cob? Okay, this is a previous year question, but this is very interesting question. Reason being, this statement is given into your NCERT and lot many students, I would say, do not pay attention to this statement, and this question was asked in PYQ. That is why I put this question in our session. Chinni, yes, Chinni. Naresh, good. Honey Reddy, good. Risagna, very good. Rupa, very good. Varshini, very good. Neer, Uttej, Vandana, Naju, Kiran, perfect, my dear kids. The question is, what is the function of tassels in corn cob? What would be the function of tassels in the corn cob? This statement is given in NCERT itself, my dear kids. If you'll open sexual reproduction in flowering plant, in pollination part, you're gonna find this particular statement. That is, the function of tassels in the corn cob is basically to trap the pollen grain. And this is a previous year question also. Lot many students were not able to do it correctly because they were confused, like from where this statement has been taken up. So this is from basically SRFP, which is Sexual Reproduction in Flowering Plant. You're going to see the same statement in NCERT itself. Okay, thank you very much, Dr. Queen. And everybody, please listen to Dr. Queen. Do hit the like button and share it with all your friends, and subscribe our channel, because you will get notified about all the classes which are going to happen in next 10 days. All right. Yes, so answer of this question will be option number C, which is to trap the pollen grains. Perfect, everybody, perfect. Moving on to the next question then. This is indeed a very important concept and important question, I would say, from genetics. See, earlier the questions were more in the mathematical side. Earlier we used to get questions like from epistasis, from complementary genes, from supplementary genes. But right now, if I speak about the latest trend from genetics portion, so basically we get question, I would say more on the easy side. Reason being, sometimes scientist name has been asked. Sometimes Mendel picked how many pure breeding varieties. This simple question has been asked. Okay, Yes. Scientist name. Please don't get confused. Come on, everybody. Chinni, Ramala, Moonshine, Naresh, Maneem. Very good. Uttej, Rakshan, Naju. Perfect. Prabhavati. Very good. Kiran. Very good. Very good. The question is, experimental verification of chromosomal theory of inheritance was done by experimental verification. See, lot many students are answering Sutton and Boveri. See, Sutton and Boveri basically gave the chromosomal theory. Question is asking about the experimental verification. Mendel sir gave us very up-to-date and very renowned results, but nobody believed him. There were several reasons to it. The first one was the publications were all in the local journal. The second was he basically used all the mathematical explanations. That was the first time in the entire history that somebody from the biology background was using mathematical explanation for any concept. Okay, so people, basically the scientists from that era, were not able to find it fruitful. That is why he failed. The third reason was he was not able to give a physical proof of the factors. Although he said that factors are basically the entities which are responsible for inheritance pattern, and this is a discrete unit also. But he was not able to give any physical proof. What is factor? Is it made up of DNA? It is made up of RNA, proteins are there, nucleotides are there, nothing is there. Okay? So yes, of course, communication was a problem. Apart from the communication, there were not many other problems also. So later on, when we were able to discover microscopes in the world, so after that, when we started, there is no voice. Am I audible, kids? Am I audible, kids? Okay. Allow me 1 minute. Allow me 1 minute. No voice? Okay, 1 minute. Am I audible, kids? Am I audible? Not clear? Am I audible, kids? It is clear now? Okay. The rest of the kids, please do let me know. Is it clear now? My voice is clear now? My voice is clear now? No? Please refresh and then start the session again. Refresh. Slight disturbance. Okay. My voice is not clear? Yes, ma'am. If somebody is still facing the problem, please refresh and then... Yes, ma'am. Now, okay. Okay. So, we were discussing about the concept of chromosomal theory. Alright. So, later on, when Sutton and Boveri, they just Now it is clear. They were able to see that chromosomes are behaving in the similar way, the way Mendel has explained. Okay, so now it is okay. Okay, perfect. Let's come back to the concept. Now, they have understood that what right now we are able to find out, Mendelssohn has given the proof, Mendelssohn has found out way back in his time, okay. So that is the reason they combined all the information and gave a chromosomal theory of inheritance. Later on, experimental verification was given by T.H. Morgan sir using Drosophila. Right? So answer will be option number C. Now it is perfect. Okay, perfect. Thank you so much. Thank you. Right? Is this clear now? The funda is clear? The students who were confused between first and C option, is this clear now? Yes, ma'am. There will be no -1 in final exam. Is that a promise, everybody? There will be no -1 in final exam? Yes, that is a promise. That is a promise to you, ma'am. Yes. We will do -1 in these classes, but we will not do any -1 in our final 2024 exam. Promise, for sure. Okay, next question then. Next question is, we need to select the incorrect match. All right, these kind of questions are misleading. Incorrect match, right? So we have option 1, DNA replication. For DNA replication, we do need enzyme, that is DNA dependent DNA polymerase. Number B says transcription needs RNA dependent RNA polymerase. Number C says taminism, RNA dependent DNA polymerase. Number D says translation, we need peptide transferase enzyme in order to transfer the peptide bond. Yes, great. Naresh, Ramala, Chini, Risagna, Maneem, Rajya Lakshmi, Kiran, Dhana, Uttej, Hani Reddy, Navya, Varshini, Dr. Queen, Neer, come on. Ashrita, come on everybody. Select the incorrect match. Perfect. I can see few students are confused between B and D, my dear kids. The students who are answering option D, translation is basically formation of proteins. When we want to make proteins, we do need peptidyl transferase enzyme, which is basically transferring the peptide bond. This is correct match. The incorrect is option number B, which is for transcription. Transcription is basically we want to make RNA from the DNA. That is transcription. Yes. Formation of RNA from DNA is transcription. In order to make RNA from the DNA, the enzyme has to be DNA dependent RNA polymerase, not RNA dependent. It is for basically reverse transcription, right? So it has to be DNA dependent RNA polymerase. That is why it is incorrect match, hence correct answer. Yes, hence it is correct answer. Siri, long term, Valu, Vunera. I guess you want to ask what is the long-term course to prepare for NEET examination. So to prepare for NEET examination, the long-term course is Vedya 2020, Vedya 25, that is already live, my dear kids. So Siri and Dr. Queen and rest of the kids, you can register yourself in this batch. The seats are already limited. There is this Google form in the description box. Go and fill that Google form. You will be able to you'll be able to find out all the features of Vedya batch. All right? Yes. Come on. So answer is option number B. +4. Moving on to the next question then. Next, come on kids. Very interesting question. Which of the following is not, this is the keyword kids, keyword, not a cofactor of enzyme pyruvate dehydrogenase complex. We have PDC, pyruvate dehydrogenase complex. Okay, this complex is basically responsible for link reaction. Responsible for link reaction. Yes, Ramala, thank you very much. Chipandi Garu. See, I also know few words in Telugu, okay? You people cannot cheat with me. Yes, Chini. Yes, Moonshine. Yes, Risagna. Rama Devi, Hani Reddy, Nisha. All right, all right. So, which is not a cofactor of enzyme pyruvate dehydrogenase complex. See, if I speak about respiration chapter, so in that we have aerobic respiration. In aerobic respiration, the number one is your glycolysis, okay? In glycolysis, my one glucose molecule is participating and it is forming 2 pyruvic acid. Now, these 2 pyruvic acid are participating in the link reaction and they will make acetyl coenzyme A. So when my 2 pyruvic acid are forming 2 acetyl coenzyme A, this kind of reaction is your link reaction or transition phase or gateway pathway. The enzyme responsible for these reactions is pyruvate dehydrogenase complex, because in these reactions we need to make NADH2 number 1, we need to transfer coenzyme A number 2, we need to shift the location also, because glycolysis is in the cytoplasm. Okay, now the next set of cycle, which is matrix, which is a Krebs cycle happening into the matrix. Now there is this gap of location also from glycolysis to the Krebs cycle, which is from cytoplasm to the matrix of mitochondria. Okay, to fill this gap, we have link reaction. All right. Yes, ma'am. So, yes, everybody is correct. I can see Rakshan, I can see Rajya Reddy, Siri, Sana Angel. Yes, Rasagna, Timote, Moonshine, Chini, Ramala, Kaveri, everybody is correct. It is heme group. We do need NAD+, we do need magnesium ions, we do need coenzyme A. Then only acetyl coenzyme A will be able to form. Okay, so answer will be option number D, which is heme. Yes, yes. It would be great if you'll ask me in English. Okay. Because I can pick just few words from Telugu, like Chapandi is greeting, Garu is huge respect to somebody. Yes? Yes? Okay. Rest words you people can teach me. Okay? The correct answer of this question will be option number D, which is heme. Perfect, everybody. +4. Can I see +4s in this question? +4? +4? +4? Yes, kids? +4 in this question. All right, moving on to the next question then. +4. Great, great. Next question, kids. Next question. How many laws of inheritance did Mendel proposed, suggested based on the monohybrid cross? Please pay attention to the question. Thank you very much, my dear kids. Yes, yes. Please pay attention. Please pay attention, my dear kids. The question has asked about just the monohybrid cross. So the students who are answering option number B, because a lot of you are answering option number B. Great, 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 great. Vanama, Prabhavanti, Uttej, Naju, Dhana, Nisha, Rakshan. Great, great. Naresh, Chini. Maneem, very good. Timhote, very good. Moonshine, very good, very good. See, the question is about how many laws of inheritance did Mendel proposed based upon the monohybrid cross. So, my dear kids, if I speak about the total laws, so total there were 3 laws, okay? The first one is law of dominance, second is law of segregation, third is law of independent assortment. Now, the law of independent assortment was based upon the dihybrid crosses. Okay, if I speak about monohybrid crosses, we had 2 different laws, law of dominance and law of segregation. These 2 laws were based upon monohybrid crosses. Okay, so the answer of this question will be option number C, which is 2, not 3. Right, and these 2 are basically, the first one is the law of dominance and the second is law of segregation. Right, everybody? So answer will be option number C, which is 2. Is this clear to all my kids out there? Yes. Is this clear, everybody? Yes. Okay. Okay. Perfect. Perfect. Moving on to the next question then. Next question, kids, on your screen. +4. In the last question, we got +4, everybody. Okay. Meanwhile, please do hit the like button also. Like, share and subscribe our channel. Next one, come on. Activity of an enzyme can be affected by a change in the condition which can alter. We can modulate the activity of enzyme can be affected by a change in conditions which can alter what? Nature of enzyme, kids, secondary structure, tertiary or quaternary structure. Come on. Great, Sana, Uttej, Kiran, Rakshan, Dhana, Vanama, Nisha, Dr. Queen, Moonshine, Rasagna, Chini. Come on, everybody. It is your tertiary structure. Yes, Thanuj Kumar, perfect answer, everybody. It is a tertiary structure. Everybody is correct in this question. Please, Dilip Kumar, a great answer. Plus for everybody in this question, plus 4. And let's move to the next question then. Next one, kids. In which of the following phase proteins are synthesized in preparation for mitosis while cell growth continues? Cell growth is there and still we are into the process of making proteins. Dhana. Next question. Ramala Kaveri, great answer. Divya, great answer. Dilip Kumar, Dhana, please pay attention and think twice. Long term ki Intha. Dr. Queen, Intha will be told to you by filling the Google form which is in the description box, okay? If you will fill the description box Google form, then all these queries will be taken care of. Yes? Great. Renuka, Uttej, very nice. Moonshine, very nice. So in which of the following phase proteins are synthesized in preparation for mitosis while the cell growth continues? Those of you who were answering S phase, my dear kids, S phase basically, in S phase, my cell is going for DNA replication, and specifically the DNA amount has been increased. DNA amount will be doubled. Okay, supposingly initially in my parent cell the amount is 2C. So in the S phase the amount will be 4C. That will be exact double. But if I speak about number of chromosomes, the chromosomes will remain same. So if my cell is having 2n number of chromosome, it will remain 2n in the S phase, okay? It will not be 4n. So please pay attention. In S phase, number of chromosomes will always remain same. Just the DNA amount will be doubled, all right? So the question is asking about the protein. So not in the S phase. G0 phase is the inactive stage, quiescent stage. In the G1, of course, organelles are being duplicated. In the G2 stage, the rest of the proteins like tubulin protein will be formed, okay? Answer will be option number C, which is G2. Give yourself a pat in the back, everybody, and a +4. And a +4. Yes, Ramala, perfect. Chromosomes will remain same. Very important line. From the S phase, very important line. Yes, Navya, chromosome number will not change at all. +4, everybody, give yourself a pat in the back, and +4, +4, great. Next question on your screen. Next question, kids. How many are associated with G0 stage? Yes, G0 stage is the one which is also called as quiescent stage. How many features are associated with the G0 stage? A cell exit of G1 phase, metabolically active stage, the cell will be in metabolic active stage. There is no proliferation and suspended stage of cell cycle. Divya, Ramala, Rasagna, very good. Rajya Lakshmi, very good. Renuka, perfect. Dhana, very good. Uttej, Dilip, Neer, Rakshan, Kiran, So fast answer of this question. Very good, everybody. Moonshine Chini. Very good, very good, very good. It is option number D. 1, 2, 3, 4. All the 4 statements are correct. See, if let's say my cell doesn't want to go for division, then it would like to exit the cell cycle, and this exit can happen after G1 phase. Okay? Your cell will decide to go into the G0 phase once it exits from the G1 phase. In this phase, my cell will be metabolically active, all right? Although there is no proliferation, but cell is metabolically active. And it is suspended stage of the cell cycle. So answer is option number D. 1, 2, 3, 4, 4 statements are correct, all right? Varshini, ma'am, I'm scoring only 250 in biology. Can I boot my score to 340 in biology by analyzing my mistakes? Yes, of course, Varshini. See, if you are at 250 in biology, Varshini, let's just be in the honest talk first of all. 250 is less marks, my dear kid, because biology is your core subject. You need to increase your marks in biology first of all, okay? And in this time, wherein we are left with just 8 to 9 days, we can increase our marks just by analyzing our mistakes. All right. So please do appear for all the sessions, analyze all these questions, analyze all these concepts which are there. I'm trying my level best to, make you aware about the concept in the deep, but just after the session also you can open up your NCERT and try to revise the concepts which are there in all these questions, okay? By revising those concepts, the concepts will be retained in your brain and you'll be able to solve questions based upon those particular concepts, okay? That way, you will surely increase your marks. Surely you will increase your marks, okay? Most welcome, my dear kids. Okay, moving on to the next question. +4 for this question. Next question, kid. 2 nucleotides are linked through. Okay, this is your last question. This is your homework question. This is your homework question in the last class. Yesterday's class. Just 2 students answered the question, okay? So this is your homework question. Please write the answer of this question in the comment section. I will personally see comments in the comment section. Please answer in the comment section. Let me take the doubt. If you have any doubt, please mention it in the comment section. Dhana is having a doubt. Ma'am, I'm not able to do problems in genetics. Dhana, see, if I speak about the latest trend, so there are no mathematical questions which are being asked from the genetics portion. Genetics is comparatively easy now. Examiner will ask you about the scientist name. Examiner will ask you about the very simple information from the genetics, like the examples of pedigree analysis, examples from the disorders, all right, scientists and their discoveries. So please be thorough with all these concepts. Incomplete dominance example, Snapdragon is responsible for which particular phenomena. From codominance, you might expect a question, okay? Not a problem. First of all, remove this stigma from your head that the genetics is such a big problem. Not at all. Genetics is such a beautiful chapter. From entire botany, I would say genetics is the simplest one, right? So just remove this stigma that you are not able to do. You are only able to do it, okay? Stand in front of mirror and speak to yourself that you are surely able to do it. Done? Okay. NP Bhargav, ma'am, biotechnology is big problem. Again, I will say the same thing. From biotechnology, the questions are little on the conceptual side. Okay, let me give you one analysis. Basically, if I speak about last 3 years, so in the NEET examination, the application part of the biology, we are expecting more questions from the application part of biology. So whether you like it or not, genetics is having maximum weightage, okay? Maximum questions will be from genetics only. Last year in 2023, from molecular basis chapter itself, there were 8 questions. 8 questions from one chapter. Okay, so you cannot avoid the chapters of genetics at all, first of all, okay? So it is having maximum weightage. Number 2, cytology is having, cytology is also on the application part. Biotechnology is also on the application part. So the number of questions will be more from these areas, be it genetics, be it cytology, be it biotechnology. If I speak about conventional botany, which is morphology, which is anatomy, so over last 3-4 years, the number of questions are basically reduced from these particular areas. So please, be really smart in your studies, and if your biotechnology is weak, or let's say your molecular basis chapter is weak, so this is the time when you should focus on these chapters for sure. Okay. Ma'am, molecular basis, yes. May I know which topic will have? Dhana, in these sessions, the questions are from entire botany. There is no bar from entire 11th, from entire 12th. Okay. Reduced portion. My dear kid, see, according to the latest NMC circular, lot many chapters have been reduced from your syllabus. Okay, if I speak about botany, transport in plants, mineral nutrition, there are certain topics from plant growth and development, some topics from living world, some topic from plant kingdom. These have been removed. Okay, so basically we do not need to study these topics, but this is the educated guess of mine, purely mine. Since we have section A, fixed 35 questions, section B, 15 questions, out of which we are liable to do just 10 questions. So those extra 5 questions, we have bandwidth of those extra 5 questions. We might expect questions in that extra portion. Okay, so the question from the deleted portion can be there, but that will be in the extra portion only. So if you will be focused enough, you will be focusing on the syllabus which is there with you, then for sure you will be able to achieve 360. Okay. Thank you very much, Uttej. I will really love to see your comments in the comment section also, just after the session. Ma'am, I'm getting 300. How can I increase? Please help me. Bhavani Prasad, see, I do not have any magic wand that can increase your marks, first of all. You are the only one who can increase your marks. And you can increase your marks just by analyzing the mock test paper, just by revising your concepts which are there. So revision and analysis. These two are the key important areas where you should work for sure. Okay, Bhavani Prasad, try it out. Okay, try it out. Trust me, you're gonna get more marks from 300 for sure. That I can say. Ma'am, I can score 300 to 330. So how can I move my score to 350 in biology? Same thing I will say to you, Risagna. You can surely move your marks first of all. Okay, remove this stigma that you cannot do it. You can do it, of course. And secondly, please analyze, please revise, revision, analyzing, revision, analyzing. These are the two things. Okay. How many questions we can expect from plant physiology? NP Bhargav, if I speak about previous years, so there used to be question from transport in plant, 1 or 2 question, from mineral nutrition, 1 or 2 question, and from plant growth and development also, we used to get question from vernalization, from photoperiodism. So we have bandwidth of 4 to 5 questions that will be divided into photosynthesis and respiration for sure. Okay, so we might expect more questions from photosynthesis and respiration this year. Alright everybody, so we'll see you in the next class. The next class will be there, that will be of chemistry by Pawan Kasu sir. I will see you tomorrow, same time, with another 25 amazing question, and thank you very much for having me. Please do hit the like button if you like the session and share it with all your friends. I will be live tomorrow, same time. See you all live. Till then, be happy, be healthy, take care of yourself. Bye kids.